Okay. Uh, yeah, thank you so much. Thank you. Thank you. Uh, okay. Okay, should be okay. Okay, thank you. Thanks. Thank you. Thanks. Okay. Okay. Oh, okay. Okay, I will. Okay. Uh, Hello, everybody. Just checking. Zoom people, can you hear me? Uh, okay.
Yes.
Yes. Okay. Thank you. Uh, oh, that's well, a little too dark. I mean, it's not a movie or something. You don't want. Okay. Uh, okay. So this is welcome to the second lecture, Fundamentals B. And so Dr. Freudenreich, I think, spent a couple minutes about what the exam looks like. So just again, there will be a midterm, there will be a final exam, each one 70 questions, multiple choice in exam soft. I hope you familiarize with exam soft. And there will be also some people ask if there's an overlap. No, there are 7 different lectures like mine here in the first part and 7 different lectures in the second part. Midterm, final exam are 2 different exams. They don't overlap or anything. And for the grading, we will see at the end how people do. And so usually it's like 70 points, 70 points. And so we have equal weight. And okay. If there are any questions, you can just raise your hands, or Zoom people, you can also, it's better Zoom people if you ask a question. I will not look in the message box always. Okay, so today we talk about mitochondria and secretion pathway. And okay, I need to, okay. So this is like an objective slide, may help you to organize your studying efficiency. Is also what I want to say for the studying is there's a lot of material. So what I did when I was student, I was splitting up this in small parts, like for example, 5 slides. You cover the slide except the subject line, and then you memorize, okay, what's there? What do you need for import in mitochondria? You can also write down with a pen on it. This is active learning and so. And you go 5 slides, go to the beginning, again 5 slides. Then you go next 5 and repeat this and go back 1 to 10. Some people they just may flip through. I'm not this person, and they have like a photographic memory. I don't have. So this helped me to store all this information in my brain. So, okay. Okay, so this is objective slides, but you also want to go like to the books like Molecular Biology of the Cell, for example, Alberts, and so. There will be 2 parts today, mitochondria, and we will dive into secretion pathway. This is the second part. Sorry. Oops. So this. Okay. You heard already in Dr. Freudenreich's lecture that the eukaryotic cell contains multiple organelles. Just to repeat it, there is there would be the nucleus having two membranes storing the chromosomes, genetic material, surrounded by endoplasmic reticulum. That's another organelle. We will hear about secretion pathway, endoplasmic reticulum. Also the Golgi apparatus. This is also in cell membrane part of the secretion pathway when proteins move from the endoplasmic reticulum to the plasma membrane. There are lysosomes. They are degrading proteins that are damaged, not needed. Secretion vesicles are moving in between, for example, endoplasmic reticulum and the Golgi, within the Golgi, going to the plasma membrane. So transport of the proteins goes by secretory vesicles. Okay, so first part, what we talk about is mitochondria, this potato-shaped structure. And mitochondria form a dynamic network. I think also you will see when Dr. Caroline Suzuki will talk about membranes. And so it's constantly changing shape. There are vesicles, there are tubular structures. Those are moving around. And I just say it also now, mitochondria is a powerhouse of the cell, providing energy. So you will need that in, for example, nerve cells, brain, heart, so for the heartbeat, contraction. And so that's very energy demanding. And mitochondria, they produce this energy and they have to be there where the energy is needed. So therefore, you have this dynamic network constantly changing the shape. Okay, so, oops. And there are also other networks in the cell with regards to vesicles and organelles. So that's endoplasmic reticulum would be another network which looks a little bit different. So this is also surrounding the nucleus. Okay, so coming back to the mitochondria, you may want to memorize they have, in fact, two membranes, not just one. So there is an outer membrane, there's an inner membrane. There would be a, okay, there would be an space, intermembrane space in between this outer and inner membrane. So this is like this kind of wrinkle structure here, where the outer membrane is more or less smooth. There are contact sites between the outer and inner membrane. We come back for this protein transport into mitochondria. These contact sites play an important part in this process. And within the mitochondria, there is this space is called matrix. And and crista is called this kind of wrinkle-looking appearance of the inner membrane. This is called crista. When we take a moment to look at the lipid, the fat distribution, and the membrane, sorry, protein distribution. You can see outer membrane is about half lipid. So lipid would be phospholipid, so what forms the membrane. Also, Dr. Suzuki will go into very much detail about this. But besides the membrane, there are proteins embedded in the membrane. So this is about half protein and fat component. This is about 6% of total mitochondrial proteins. When we look at inner membrane, we have much more proteins there, 80%. So 80% protein, only 20% lipids. So it's way more of proteins. Inner membrane, we have an important feature of this. It's a membrane potential. So there is a difference in the charge when we look in the matrix versus the intermembrane space. I will come in a minute about this. So membrane potential is also important for energy providing, when we talk about respiration, oxidative phosphorylation. So that's the important part. Intermembrane space has little proteins, 6%. Majority, you can see matrix contains about 2/3 of all mitochondrial proteins can be found in the matrix. So this is this inner space here, 2/3 of the proteins. So there are numerous functions associated with the mitochondria. Of course, I told you there is mitochondria as a powerhouse of the cell, and they have this respiration and oxidative phosphorylation. These processes provide cellular energy, but there are also other numerous functions associated. One would be, for example, protein translocation. About 99% of the proteins would come from outside, encoded in the nucleus, synthesized in the cytosol, and transported into mitochondria. There's only a little portion of the proteins are synthesized in the mitochondria. So these are here, mitochondria contain their own genetic material, mitochondrial DNA. It's very small. And so, thank you. Thank you. Thank you so much. So they have their own DNA, they have their own tRNAs, ribosomes. So a small portion of the proteins can be synthesized directly within the matrix. And so I come also about this topic. Some other things, I told you mitochondria form a dynamic network. So there are processes associated with the fusion and fission of vesicles, forming these tubular structures. And so there is this process here has some proteins involved. And apoptosis, programmed cell death, that's another important feature. And mitochondria are much involved in this process. I come just 1-2 slides about this. So, okay, respiration and oxidative phosphorylation, that's where we get, say, cellular energy, ATP. Again, mitochondria provides the energy, so they are powerhouse of the cell. Usually we have in our food, we talk about, for example, carbohydrates and fatty acids. So they would provide energy, and within, say, there are some processes that would digest, say, carbohydrates and fatty acids and provide this to the mitochondria, so they can easily generate the energy. In mitochondria, we have, say, respiration. That's basically to transport electrons. I come in a minute to this, but respiration coupled with the oxidative phosphorylation, that's a process that produces the ATP. And these two, respiration and the phosphorylation, are coupled to each other. So there is a first part, I think in Funds A, you learned already with Dr. Rogers, Melissa Rogers, glycolysis. And so glucose, which is the number one source, carbon source, is digested into smaller parts during glycolysis. And glucose, as you can see, has 6 carbon atoms, and this will be split into 2 equal-sized parts. It's called pyruvate. And so this has only 3 carbon atoms. So there will be 1 glucose ends up as 2 pyruvate molecules. And this pyruvate is incorporated into mitochondria and further on degraded in the citric acid cycle to form, let's call it, energy equivalents. Let's call it this. Okay, so glycolysis is this part. You don't need to memorize this, but what you may want to memorize is glycolysis, starting off with glucose. The experiences multiple phosphorylation steps in the first part using ATP. Then it's split up in 2 equal parts. What I told you from is containing 6 carbon units, so ending up with 2 times 3 with 3 carbon units. And in this case, we would, so here we would use ATP in the first part. In the second part, we would generate ATP, and we would end up with 2 molecules of the pyruvate. So all what you may want to know is there are some phosphorylation steps, and in the second part, you would actually produce ATP. Important is 1 glucose will generate 2 molecules of the pyruvate. So you go from a 6 carbon unit to a 3 carbon unit, and you have 2 molecules of this. So this is a little bit a busy slide, but it's an overview. It's a scheme what you would see with respect to respiration and oxidative phosphorylation in mitochondria. So we just talked about glycolysis, generation of the pyruvate, coming from one glucose to two pyruvates. And this will be incorporated, transported into mitochondria, and will be introduced to the citric acid cycle. At the same time, fatty acids, part of, for example, phospholipids, or like a glycerol connected with three fatty acids, for example, they would be incorporated, also transported into mitochondria, and can also serve as an energy source. I just mentioned this and leave it like this. I only talk about the glucose part. So the pyruvate will be incorporated in the citric acid cycle, and citric acid cycle are numerous steps to degrade the original pyruvate, and it will generate energy equivalence. So this is called this NADH, or electrons are transferred to this compound seen on the left side here. So this is called nicotine amide adenine dinucleotide. So you have say here say adenine dinucleotide, and there's this nicotine amide. So this compound allows to take up electrons, and this taking up of electrons will increase the energy level. And this compound then transfers electrons, so energy, to the respiration complexes. I will come, I think, next slide. Sorry. No, sorry. So these complexes here, they just pump protons from the matrix to the inner membrane space. And they use these energy from the electrons, they use this for the pumping activity. Okay, so this is what's happening during respiration. At the end of respiration, these are multiple protein complexes. So at the end, you would see a transfer of electrons to an oxygen, and you would form plain water. So this is what's respiration called. And at the same time, these protons here, these protons here will be pumped back into the matrix by the oxidative phosphorylation complex. This protein is pumping proteins into the matrix, and that helps to generate ATP from ADP. So overall, what you want to memorize is you incorporate oxygen. Oxygen will be converted to water. You provide ADP, which is coming from outside, and is converted to cellular energy, ATP. So this is then released from the mitochondria and needed for energy requiring processes. And third, you would see carbon dioxide will be formed from the citric acid cycle, and this will also be released. So this is some kind of like a balance. Yeah. Okay, so this is also, I think you heard about in Fundamentals A about this. The TCA cycle helps to degrade the pyruvate that comes from the glycolysis, and it will transfer energy equivalence to this NAD molecule. What I just told you before, it will take up electrons, and these electrons are then transferred to the respiration complexes. So there are 3 NADH molecules generated per per glucose. There would be per cycle, sorry, per cycle. There would be one also GTP, that's another way like ATP or GTP. And a flavin, FADH, is another way to transfer electrons to the respiratory complexes. And there would be carbon dioxide will also be released. So you want to memorize what's generated per cycle in the citric acid processes. When we go a little more in detail, respiratory chain reaction, these are uh, multi-protein complexes, and these complexes are so most of these are generated in the nucleus, encoded in the nucleus, translated in the cytosol, then transported into mitochondria. A small portion of these complexes are transcribed by or encoded by mitochondrial DNA. So there would be complex 1, 3, 4, just roughly saying. There would be other components like ubiquinone is not a protein, but it transfers electrons, for example, from complex 1 to complex 3. It's sitting in the membrane, and as I said, it transfers electrons, but it's not a protein. And then there is cytochrome C, which is sitting on the outer side of the inner mitochondrial membrane. It's in the intermembrane space, but attached to the inner membrane, and also this transferring electrons. So we have multiple complexes which transfer electrons from one complex to the next, and those are coming from this NADH, which is transferring energy equivalence, so these electrons. And at the end, you would form water when you use oxygen. So all these complexes, like 1, 3, 4, they would pump protons from the matrix to the intermembrane space. So this means the intermembrane space will become more positively charged, whereas the matrix will become negatively charged. So there is a proton gradient established between matrix and intermembrane space. Okay, so this is about respiratory chain reaction. yeah, again, ubiquinone is not a protein. Uh, and so here, respiratory chain reaction. And these protons that accumulate in the intermembrane space are pumped back in the matrix uh, by the ATP synthase. So this protein complex also sitting in the inner membrane, this protein complex generates ATP, the cellular energy, by using ADP. So adenosine diphosphate has two phosphates, so there is a third phosphate because it's adenosine triphosphate, and this would be the energy providing compound. Okay, so and this all these processes is coupled to this pumping of the protons from the intermembrane space to the matrix. Okay, so this is just an overview what you would get in terms of ATP derived from one molecule of glucose. And so you have, for example, glycolysis, you would have, for example, two ATP generated. So you remember there were phosphorylation steps. So there are four ATPs generated, but two ATPs are used. So the net outcome would be just two ATP would be generated. The same with two electron transferring units, this NADH would be generated. Then, yeah, you would go from pyruvate to 2-acetyl coenzyme A. That's what's incorporated in the 3-carbon cycle or citric acid cycle. And now everything goes times 2 because you have one glucose will generate two pyruvates. So everything is times 2. So at the end, I mean, this is just like a way so there would be two, this flavin, FADH, 6 NADH, 2 GTP would be generated, for example. So at the end, you would generate about 30 ATPs. So in literature, this can fluctuate. Also, some say 38, 32. So it depends on how you do these biochemical reactions to come to these numbers. But in overall, it's like in the range of 30 ATP molecules derived from one glucose. So you want to memorize this number, maybe might be important. So this before respiration, oxidative phosphorylation is everything when you use oxygen. So there would also be a situation where oxygen would not be present or would be limiting. And so glucose would then would be degraded in glycolysis to pyruvate, but it would not be incorporated into mitochondria. It would be further converted to lactate. And so, this is called anaerobic glycolysis. And this actually happens when you do like a workout in the gym, and you would you would do more than you actually want to do, and you get muscle sore. And that's actually this accumulation of the lactate, because there would be a limitation of oxygen in the muscles, so you would not produce energy in the mitochondria. Okay, so this is without the oxygen, but there is also an effect that you can observe in dividing cells, proliferating tissue, or in cancer cells. And this is called Warburg. This person found this the first time, that tumor cells, although there are functional mitochondria, they would generate majority of the cellular energy, so ATP, during glycolysis. So they would not use mitochondria. They would everything would be based on basically on the glycolysis level. So I mean, you can read up on this. I just want to mention this. There is this pathway in tumor cells, also in proliferating cells, because most cells, like when you look at, for example, I'm just saying cardiac cells, for example, are not proliferating, for example, or neurons would be end differentiated, they would not divide. But proliferating cells, they may use this pathway where most of the energy or lots of energy will be provided by glycolysis. Okay, so well, this is just what I said before, and yeah, this is now we are heading to say how mitochondria are generated. So I told you already there are almost 99% of the proteins in mitochondria are encoded in the nucleus, in the chromosomes, and so there's transcription in the nucleus, there is RNA, messenger RNA transported to the cytosol, and then by ribosomes, you would see translation, and these proteins that are generated are imported into mitochondria. So these are 99%. And this can, there's a range, for example, budding yeast, baker's yeast, could be 500 to 800 proteins. Human heart, 1,000 to 1,500, depending on various situations. For example, if a lot of energy is required, or for example, normal cells, tumor cells. So there could be like a variation of the amount of proteins. Okay, so protein translocation, folding degradation. So that's for most of the proteins that are coming into mitochondria, but there are only a tiny, a handful, more than that, couple proteins encoded by mitochondrial DNA, and there are also tRNAs and ribosomal RNAs. So they have their own here ribosomes needed for translation. So when we talk about mitochondrial DNA, this is more or less looking like what you learn with bacterial DNA. It's, first of all, it's circular, and it's attached to the inner membrane on the matrix side. So this is reminiscent of when you look at bacterial DNA, is also attached to the membrane. It would also be circular DNA, So it's very similar. When we talk about chromosomes, there would be a fixed number of chromosomes in the nucleus. In contrast, when we talk about mitochondrial DNA, there could be a variation. Some cell may have a few copies of mitochondrial DNA. Other cells may have tons of, lots of copies. So also this may change with energy requirements. So if more energy is required, so you would see probably an increase in this number of mitochondrial DNA molecules. Okay. When we look at the structure, human mitochondrial DNA is quite small, 16 kilobase. It looks like a plasmid DNA. And so there is just one site in this plasmid where it starts with a DNA duplication process, replication, and also transcription, where you form, say, messenger RNA. There would be like one messenger RNA would be formed, and that would be split up into smaller parts. So there would be, for example, 13 proteins of the respiration complexes would be encoded by mitochondrial DNA. The other parts of the respiration complexes are all encoded in the nucleus. There would be ribosomal RNAs, so that's part of the ribosomes, and there would be numerous transfer RNAs. Those have a little different codon usage, this triple codon, like for example, ATG for methionine. So some of these codons may have a different triple codon compared to what you would see for the nuclear genes. So just to mention there is this difference. Okay, so you want to memorize what mitochondrial DNA contains. So say couple proteins of the respiration cycle, ribosomal RNAs, tRNAs. So mitochondrial DNA, or let's say mutations, and so are associated with numerous disease. So this is known about maybe 20, 25 years or so. There could be at the bottom you see here point mutations. There could be small portions of the mitochondrial DNA could be deleted, or mitochondrial DNA could be totally missing. So it could be reduced to very small number of mitochondrial DNA molecules. So there, you can see in various cancers, like point mutations in mitochondrial DNA. I remember there was a science paper describing a point mutation in a specific tRNA gene in mitochondrial DNA that goes along with metastasis, for example. So they could show this point mutation in one tRNA is linked to inducing metastasis, spreading of tumor cells in the body. Other disease would be cardiovascular disease. Well, heart needs lots of energy. So if there's some failure in mitochondrial DNA, you cannot provide this energy. So you have some kind of progressive heart failure. Also, diabetes is associated with some mutations. Neurodegenerative disease like Alzheimer, Parkinson, for example, loss of neuron function. Neurons need also a lot of energy, so a lot of mitochondria. And as I told you at the beginning, there's a dynamic network, say nerve cells, say axons, I think, call it X. So nerve cells are huge. So mitochondria move along. So if you lose this ability and say ability to generate energy, so you would see progression of Alzheimer's, Parkinson, for example. Premature aging syndrome, it was observed that people with advanced age, they would lose a certain portion in mitochondrial DNA. So as I told you, there could be huge variation in the number of mitochondrial DNA molecules, so going from 100 to a couple thousand. So you would see like a threshold level normal full-sized mitochondrial DNA versus those with a truncation. Because if it's all molecules have the truncation, you would see some respiration proteins would be missing, so that wouldn't make sense. So it's like a threshold level that cells may, for example, choose to undergo cell death if energy cannot be provided. So again, premature aging syndrome is associated with some specific deletions. Depletion of mitochondrial DNA, that I just want to mention, could be, for example, with some drug treatments. For example, there was the original drug to treat HIV. This was ACT, had a side effect of removing mitochondrial DNA. And so as a side effect, some patients experience like heart failure. And so they came back to this, that the drug affects propagation of the mitochondrial DNA. So it's basically depleted from the cells. So you see a lot of different ways, point mutations, deletions, depletions. So, okay. So coming to say, this was mitochondrial DNA, just a couple proteins are encoded. Most of the proteins that you find in mitochondria are coming from outside. So coming from the nucleus, nuclear encoded genes. And so you would see messenger RNA is transported to the cytosol, translated by cytosolic ribosomes, and then a fully synthesized protein will be imported into mitochondria. There are also other organelles shown here. For example, besides mitochondria, chloroplast, for example, peroxisomes. Endoplasmic reticulum is a different way. I come in the second part, secretion. And so there is a different way to import proteins in this organelle. So usually, and proteins can also be imported into, of course, nucleus through nuclear pores. And so, okay, so when proteins are imported into, say, mitochondria, there would be, out of logic, you would see 4 different locations. So there would be the matrix proteins, There could be, say, outer membrane protein, inner membrane protein, or it could be somewhere in between. This requires different targeting signals. So you have to tell the protein where it should go. So that's an important slide. So the default pathway is when proteins are ending up in the matrix. To do so, the proteins contain a specific signal at the beginning of the protein. So N-terminal part is the beginning, C-terminus would be the end of the protein. That's how it's translated, produced by the ribosome. So this small portion at the beginning, called mitochondrial targeting signal, is a stretch of different amino acids. You see, for example, arginine, which is positively charged, lysine, the same positively charged amino acids. There are some in between. It's not very clear what exactly the signal would be. But when you look at the structure of the signal sequence, you can see like a distinct feature. And so it, first of all, it forms like a helix structure here, an alpha helix. So, and when you look here, so there's this blue arrow. You look from the front here, you would see like positively charged amino acids can be found on one side, and the hydrophobic amino acids or other amino acids would be located on the opposite side. So here, positively charged amino acids, arginine, lysine, is on one side, hydrophobic amino acids on the opposite side of the helix. So you want to memorize this very careful. So I know. I hope you understand what I. Okay. Again, hydrophobic on one side, positively charged on the opposite side. So there would be, so this would be the default pathway. There could be 20 to 80 amino acids. Some of these signal sequences may be cleaved off after import. Forms amphipathic alpha helix. So, and most of the proteins would contain this signal sequence for the import at the N-terminus. So this would be at the beginning of the protein. And so, yeah. And if a protein is targeted to different locations, like the inner membrane space or the inner membrane, for example, there would be additional sequences within the protein that may do this targeting to these places. So when a protein is going to be imported into mitochondria, there are two major complexes that play a role in this. So it's called TOM and TIM for translocase of the outer membrane, OM, and TIM, the translocase of the inner membrane, this IM. So these are located in the outer membrane and inner membrane, and they are located in a way that they are attached in close proximity at, it's called contact sites. So, yeah, okay, I come in a minute to this. Oops, so yeah, so there would be basically couple, actually, I think 3, 4 different processes with the import. Number one would be the protein that's going to be imported needs to be unfolded. So proteins are synthesized by ribosomes in the cytosol, and during this process, the protein itself would fold up like a spaghetti ball, would fold up. It's also protein itself has hydrophobic amino acids and hydrophilic charged amino acids. So say, for example, like lysine, arginine, for example, leucine, for example, are charged amino acids. So they would sit more on the surface and interact with the water environment. Whereas hydrophobic amino acids, they would be more internal. In order to be translocated through the membranes, these proteins need to be unfolded. So they would not go the spaghetti ball by itself just going through these membranes. They would look more like a string and go through these translocation channels. So to help with this unfolding, you need, here it's just called HS, HSC or HSP. These are called heat shock proteins. They help to unfold these proteins. And you can also see those unfolding requires energy. So this is, for example, one example where you need energy, ATP, for this unfolding. Once the protein is unfolded, the signal sequence will interact with the receptor in the outer membrane. Receptor recognizes specifically this mitochondrial targeting signal, what I was just talking before. And so in the next step, you will see like an insertion of the targeting signal into these translocation channels. And then it will be followed by dragging or pulling the proteins through the outer translocation channel and the inner translocation channel. So these here would be these two protein complexes are in close proximity at the contact sites. I think this, no, okay, later. And you would pull this protein into the matrix, and in the matrix you have this protein gradient that helps to pull the protein into the matrix, and you have also heat shock proteins that will help to pull the protein through the translocation channel. I mean, just highlight should be here. One part is that the protein attaches to the correct membrane, so protein is going to mitochondria. And in the second part would be insertion and translocation of the proteins into the matrix. Let's go. So again, this first part would be using these heat shock proteins, that the proteins are first unfolded in the cytosol using also ATP, and needs to be in an unfolded conformation before import. once it's unfolded, the mitochondrial signal sequence can be recognized by the import receptor, and then it will be transferred to the import channel. This is translocase of the outer membrane, and then on further in these contact sites, we say translocase of the inner membrane. So these two here would be, I think, yeah, here. This is electron microscopic picture, and you can see here contact sites is where the membranes attach to each other. So this process here of the translocation of the protein into the matrix requires energy, requires also a heat shock protein that pulls the protein through these channels here. So you need ATP for this. And second part is the membrane potential plays an important part in this dragging or pulling process. You remember the signal sequence, the mitochondrial signal sequence has positively charged amino acids, arginine, lysine, positively. During the establishing of the membrane potential, you have to go back to respiration. Protons are pumped from the matrix to the inner membrane space. So what you create is a negatively charged environment in the matrix. And that drags or pulls the positively charged signal sequence into this environment. So it's like an electrostatic interaction, the positively charged signal sequence with the negatively charged environment of the matrix. So because there's this membrane potential. Once the protein is completely inside in the matrix, there are also heat shock proteins that help to fold it to the mature, to the active conformation. Okay, so what you need for the protein import matrix, like I said, default pathway, you need a signal sequence. The protein in the cytosol needs to be unfolded, requires heat shock proteins and ATP. And this was just in the previous slide. We need a membrane potential across the inner membrane because there would be a negatively charged environment in the matrix that will interact in an electrostatic way with the positively charged signal sequence. And you need ATP in the matrix because there are certain heat shock proteins that also pulling the protein through the translocases. So you want to memorize what's needed for this process. Okay, so there would be just 1 or 2 slides about programmed cell death. This is also associated with mitochondria. It's in fact something that's, how to say, like when a protein finds itself at a place where it shouldn't be. So for the programmed cell death, is a programmed sequence of events You can eliminate cells, for example, which are harmful or so. But you would go. It's a certain sequence, and it's also important for in development. So I think the first time it was shown in the worm C. elegans. There's about 950 cells, and they could pinpoint cells that would undergo cell death or not cell death. So it was like an important part of development for an organism to have this apoptosis process. There could be too much apoptosis, there could be too little. That's not good in either way. So excessive would be, for example, what you see in neurodegenerative disease, for example, or in some immunodeficiency syndromes. Absence of apoptosis or too little of this, for example, would benefit cancer cells to develop, to spread. So there would be, when we talk about, say, process, there would be, in fact, two phases which can be distinguished. One is an initiation phase. So there would be, for example, DNA damage. Could be usually DNA damage is repaired if there are small portions of the DNA damage. If the damage is extensive, which could be, for example, high radiation, for example, then cells would commit to this programmed cell death. There would be another part would be oxidative stress, which could either come from outside with agents that create oxidative stress. Oxidative stress means, so we talked about in the respiration, transfer of transfer of electrons to the oxygen to form plain water. But this intermediate, when electrons are transferred to the oxygen, oxygen is very reactive. It's called reactive oxygen species, or ROS, and those may interact with anything. So they could interact with proteins, DNA, RNA, membrane, other compounds. So they provide a lot of damage if they are present in excessive amounts. You need, there's also literature, you need a certain small level of ROS for normal function, but if this is an excessive amount, then we talk about oxidative stress. So if that cannot be repaired, or these ROS, reactive oxygen species, can be deactivated by some enzymes, you would see inducing of the programmed cell death. Okay, so these are two examples: DNA damage or too much DNA damage, oxidative stress. There would be ion fluctuation, would be what, for example, in cardiac cells. Cytokines would be another way. Downstream of this are events that would induce degrading the cells. So this would be, there would be, for example, proteases, nucleases would be activated, and they would help to digest the DNA, proteins, membranes, and so on. An important feature in the programmed cell death is the rupture of mitochondrial membranes. So it's called permeabilization of the outer or inner membrane, and that affects or release of proteins to an environment where it shouldn't be. So one example would be here with the cytochrome C. You remember it's present in the uh, inner membrane space attached to the inner membrane. It's an important part of the respiration. But so that's like a home base, and this cytochrome C, if that finds itself now in the cytosol where it doesn't belong, it will induce certain processes which are not planned, actually. So cytochrome C would activate caspases and would induce nuclear DNA fragmentation. So something was not planned for the cytochrome C. Other parts would be, for example, calcium release may also play in this programmed cell death. There are numerous proteins known to stabilize or destabilize the outer mitochondrial membranes. Some would be, for example, BCL2 would be, for example, stabilize outer membrane, or some other, I mean, these are well-studied, well-established proteins, so lots of known about this. So yeah, I think you will maybe hear about this with Patricio. He may, I think, more go into detail with this. Last part of this, no, not last, but just calcium release. It's another feature of mitochondria. Just very briefly, has something, also stimulates, has some connection with oxidative phosphorylation, with the ATP generation, but there is also some connection with the apoptotic cell death. And most importantly, Dr. Gopal Babu, he will talk about heart tissue, and also, I guess, the calcium that's released from mitochondria, and which is important for muscle contraction. So also this is important for yeah, mitochondrial function. So mitochondria itself, when we talk about the organelle, they were not always there in the cell. They came from something, and so it's called endosymbiontic hypothesis that mitochondria have a bacterial origin, and so a precursor cell millions of years ago, they didn't have this mitochondria, but bacteria were taken up by these precursor cells, and first the precursor cells contained all the genetic information what's needed to make this bacteria, but over the time this DNA was transferred to the nucleus, and so that's what I said, 99% majority of the mitochondrial proteins are now encoded in the nucleus, and only a couple proteins remain encoded in the mitochondria. I mean, there is also some evidence that this transfer is still going on. There are some rare disease where you could see pieces of mitochondrial DNA inserted into nuclear genes, And so this is also what people study. But overall, there's a precursor was a bacterium. Most of the DNA from this precursor bacterium transferred to the nucleus. And what you can see now in our cells, most genes are encoded in the nucleus. Most mitochondrial genes, mitochondrial proteins are encoded in the nucleus. Similar with, say, chloroplasts, just here as an example, photosynthetic prokaryotes, they produce oxygen. So they were also bacteria before, and so you would see transfer of DNA to the nucleus from the precursor chloroplasts. Okay, so coming, maybe to the second part. So secretion pathway. Are there any questions or anything or it will come later? Yes, sure.
So talk about memory potential and how that contributes to the proteins that are destined to go to the matrix to end up in the matrix, right? And it helps with pulling through.
Yes.
So these other proteins that are signaled for, say, the intermembrane space or such, they have a different.
Well, this could be like I will come maybe here. They could have a hydrophobic sequence, which is getting stuck in the membrane. That would be another way. Or there could be also another receptor, for example. Like I think what I know is default pathway is into the matrix, but if a protein goes back to the, for example, intermembrane space, it would be transported out through the inner membrane into the intermembrane space. So it would not be direct way coming from outside into the intermembrane space. It would go matrix and then going back. So that's what I know. So one way would be that there's a hydrophobic stretch of amino acids, and that will getting stuck in the membrane. So that could be. So once it's, for example, on the way into the matrix, it would get stuck in the inner membrane, and then it would not be released, for example, in the matrix. Maybe.
Yeah, I just wanted to clarify.
Yeah, it's very good. Yes. Yes.
How that change the process in which
It would be additional processes like interacting the hydrophobic stretch of amino acids would interact with the lipids in the membrane. So maybe that's okay. Okay. Let's go. Yeah. Secretion pathway. Maybe you remember Diego when he talked last week about organelles. I think he mentioned also vesicles, and so we talk here about messenger RNA, which comes from the nucleus, and it's started to be translated. But there is a little difference now that it's not completely translated. There is a little stop. The ribosome will stall basically and will float to the outer side of the endoplasmic reticulum, and then it will continue translation and translocation at the same time. So this is a difference what I talked before. Mitochondria is complete synthesis, then unfolding of the proteins, then import. In this case here, you start with the synthesis, then there is a stalling, stop of the process. The ribosome would float or would go to the outer membrane of the endoplasmic reticulum, would dock, would attach to this side, and then it would continue the import or translation and import at the same time. So this is the first step in this secretion pathway would be this import. It could be import into, say, we talk about lumen, so like the matrix, we talk here lumen of the endoplasmic reticulum, but there could also be lots of proteins could just be stuck in the membrane of the endoplasmic reticulum, similar what I was just saying with the mitochondrial proteins, which have a hydrophobic stretch, they would be stuck in the membrane. So some are either imported into the lumen, some are stuck in the membrane. Then some proteins, they would just stay in the endoplasmic reticulum. Some proteins would go to the next compartment, which is the Golgi apparatus, Golgi complex. And this movement goes within vesicles. These are little balls of lipids, something like what you get with, for example, Pfizer or say Moderna with the liposomes, with the mRNA. So it's similar principle. So you have a vesicles floating. It's not really floating, it's a direct movement going from one organelle to the next organelle. And Golgi is quite complex, have different structures, substructures, cis, medial, trans Golgi, before and after this. So, and you would end up from the Golgi, you would end up, most would go plasma membrane, proteins that are modified in the endoplasmic reticulum, in the lumen, they would find itself going outside on the surface of the plasma membrane. Some proteins which are maybe not properly folded, which are damaged, they may go to lysosomes. Lysosomes degrade proteins. And some proteins, they may even leave the cells. So those are secreted, totally removed from the cell. Okay, so this is a scheme. This is just to highlight, this is very basic knowledge, and so don't need to study this, but it just highlights there are, for example, Jim Rossman, Randy Sheckman, Tom Südhoff, they provided the basic steps to explain secretion pathway. For example, Randy Sheckman used yeast mutants to figure out the movement of proteins from the ER to the Golgi. So these yeast had some mutations that prevented, for example, movement of the vesicles from the ER to the Golgi or the other way. So and they did, the initial, very basic knowledge. So again, like with the mitochondrial protein import, it needs a signal sequence. So you see here also mitochondria with the positively charged amino acids. With the import into the ER, you will also find this more or less at the beginning of the protein. And this contains also some negatively charged and hydrophobic amino acids. And so I will come in a minute what's important about this signal. There is another signal which prevents the protein going, for example, from the ER to some other organelles. It will go back to the endoplasmic reticulum. It's called ER retention signal, and it's found at the C-terminal at the end of the protein. It's called this KDEL peptide. Okay, so these signal sequences are, so from this experiment here, are not depending on the rest of the protein. So they would function by itself, and you can show that by genetic engineering. So you would have, in fact, here you have two proteins, one protein naturally having the signal sequence, ER signal sequence, so it helps to import this protein into the lumen of the endoplasmic reticulum. You have another protein which is cytosolic and does not contain any signal sequence. So by genetic engineering, which means on the DNA level, you can now remove, for example, the signal sequence, because that's when you go back, it's a stretch of DNA, triple codons. And so you would remove this from the protein that normally can be found in the endoplasmic reticulum. And when you do so and express this protein without the signal sequence, you find suddenly this protein now in the cytosol. So this means that the signal sequence itself is the only feature that causes the protein going into the endoplasmic reticulum. There's not any other, like a 3D structure or something. It's a signal sequence that's important for the import. In the other way, the cytosolic protein, you can add this signal sequence, the same principle. On the DNA level, instead of, for example, you have ATG, the start codon, and then you would have the cytosolic protein. So you would have first the signal sequence would be encoded, and then you continue with the cytosolic protein. And now you see this protein, which is normally in the cytosol, can be found in the endoplasmic reticulum. So again, you add the signal sequence, and that makes the protein going into the organelle. So there's not any other feature on the protein that it's needed for this import. It's just the signal sequence. Okay. So coming to this process of the stalling in the translation process, I think Dr. Friedenreich maybe had this slide or similar slide. There would be three ribosomes, what you can find in the cytosol. You, for example, synthesize mitochondrial proteins. They would be synthesized, fold up, but then get unfolded. But there would also be membrane-bound ribosomes. And that's the case for the endoplasmic reticulum import. It's the same pool, what you see here. So you would start here, you would start here with the synthesis of the protein. But for the cytosolic protein, for example, you would just, it continues synthesizing, and of course there would be multiple ribosomes would dock to the same messenger RNA and would synthesize it completely. In contrast with the ER, it would start off here, but then it would get bound, attached to the outer side of the ER. And once it's attached, it will continue with the translation process and the translocation process at the same time. So that's the difference with the mitochondria protein. But importantly, it's the same pool of ribosomes, what you can use here synthesizing cytosolic protein or going to the endoplasmic reticulum. And the difference is here with the ER signal sequence. So this would be electron microscopic picture, ribosomes attached to the outer side of the ER. Before, there is another experiment that shows you need both the ER endoplasmic reticulum membranes and you need the ribosomes basically in the same reaction. So in this example here, we are using purified components. So this is, we are not talking about something like looking into the cell microscope or something, or cell extract. We are talking about having purified ribosomes. You can go to some, like a company website and buy an in vitro translation kits containing ribosomes, all components what you need, and has nothing to do with like say cellular context or so. All this is done in reaction tubes and no cells present. In the first experiment, first slide, you would use this called in vitro translation kit. In contrast to in vivo, that means when you look something directly in the cell, in the animal, for example. In vitro means you have these purified components, you synthesized proteins, and then you make a protein extraction. So you have just the proteins present, and these proteins would contain this signal sequence for the import into endoplasmic reticulum. But with the differences, you have no ribosomes here present, and then you would add microsome membrane. And in this case, the completely synthesized proteins with the signal sequence remain outside. They are not going to be imported. So there has to be some feature connected to this with the ribosomes. So therefore, in the second experiment, you have ribosomes, the messenger RNA, so this in vitro translation kit, plus the microsomes in the same tube. Oh, maybe I should also, microsomes are, so this stands for endoplasmic reticulum. You can isolate anything like nucleus, you can isolate mitochondria, you can do Golgi apparatus and endoplasmic reticulum. But when you uh, open cells, gentle is you open it so you wouldn't disrupt completely organelles. And you would say endoplasmic reticulum, which is more or less like a flat, like a structure. You remember this was surrounding the nucleus and forming like layers of membranes, would fall apart and would form vesicles, would form like these balls. But those have the same property as the layers that you can find naturally in the cell. So it's the same properties of endoplasmic reticulum, but just in this case, it's forming like little lipid balls. So in this case, we have the microsomes plus the ribosomes all in the same tube. And now we can see protein is translated and translocated at the same time successfully into the microsomes. So conclusion of this, you need both ribosomes and the microsomes in the same environment. So in the same tube, you cannot separate this like before the slide. Synthesis of the protein, complete synthesis, you remove the ribosomes, then you have the microsomes present. That will not help you to get it imported. You need both ribosomes and the membranes of the endoplasmic reticulum in the same environment. So that it's a translational, translocational process. Okay, so this slide would explain why this is like this, because we would see here, we would start off with the ribosome starts with the translation. Once the signal sequence, so this is this little red stretch here, would emerge, would come to the surface of the ribosome, there is a protein particle being attached to this signal sequence and at the same time attached to the ribosome. So there are two different contacts. One is it's called signal recognition particle, will interact with the signal sequence and will interact with the ribosome. So that causes to stall the translation process. So it will stop here, and then the ribosome would float or would look for this outer side of the endoplasmic reticulum. And once the signal recognition particle would interact with the SRP receptor in the membrane. So this receptor recognizes the SRP on the ribosome. Once this happens, the signal recognition particle will be released, okay? And the signal sequence will be inserted in the translocation channel, and translation and translocation continue. So the key part is this signal recognition particle. So you want to memorize what it does, where it binds, and where it's needed. So it will stall for some time translation, and it binds to the ribosome as well to the signal sequence. Once it will interact the SRP with its receptor, the SRP will be released and the ribosome docks to the translocation channel, and translation and translocation continue at the same time. Okay, so study this very careful. So this is like a default pathway would be like complete translocation, signal sequence for most proteins will be cleaved off by a peptidase. And so in the mature protein will fold up in the lumen of the endoplasmic reticulum. So for this would be, I mean, the most basic way, but in reality, there would be multiple situations, like there could be proteins getting stuck in the membrane. And this is also what I was talking before with the mitochondrial proteins that are in the membrane. They have a membrane spanning domain which contains hydrophobic amino acids, so they would interact with the hydrophobic nature of the membrane. As I told you, Dr. Caroline Suzuki will go into detail, phospholipids, fatty acids, and all this. So these hydrophobic amino acids, they would interact with the fatty acids. So, and that causes to getting the proteins stuck in the membrane. There could be different orientation of this, like you can see here, N-terminal part inside or outside, different orientation. Many protein complexes that are sitting, for example, in ER, plasma membrane, they would pump, for example, certain ions through the membrane. They would look like this here with multiple membrane spanning domains. There would be, for example, here voltage gated calcium channels, for example. Don't need to memorize this. It just highlights there are lots of different structure possibilities. Or for example, here there would be like a lipid, phospholipid linked protein. So that's connected with the membrane via say a lipid, not via a protein. So okay. So just to highlight different possibilities for membrane proteins. This is more in detail here. Once the translocation process is in progress, there would be like a hydrophobic stretch of amino acids, and that would interact with the hydrophobic nature of the membrane. So therefore, this membrane spanning domain would stop in the membrane, and then you would create a protein that's partially outside and inside in the ER and having a membrane spanning domain. So this is overall was how proteins are getting to the ER, how they are getting into the ER, how they are folded. The folding process itself may require some additional help, chaperones, which bind to the proteins that are getting folded. If they would not be properly folded, for example, they would even be immediately degraded. So there would not be a pathway to the lysosomes. It would be some connection with the endoplasmic reticulum directly, the degradation process. So chaperones that help to fold the protein, or also recognize misfolded proteins, so there are different ways. And another way to preserve structure or to introduce structure is by forming protein disulfide bonds. So there is the amino acid cysteine, yes, cysteine, which has an SH group, and there could be, in this process here, there could be two cysteine amino acids which are far apart. They could, for example, a protein may form a loop, but at the contact site, these cysteine will interact and will form this disulfide bond. So this helps to make the protein more stable and create a certain structure. So protein disulfide isomerase helps to make interaction of cysteine amino acids, which could be hundreds of amino acids apart. So, but proteins, once they are synthesized, they may form loops, for example. And so at the base of the loop, you would see, for example, interaction of these two cysteines, for example. Okay, quality control, yeah, I mentioned this. Okay, so disulfide bonds then helping to interact, now how to say, to fold. And another way what's with protein modification is adding a sugar component. So during the translocation process, at an asparagine amino acid, a sugar component will be added, and it's called oligosaccharide, and it contains multiple different sugars like glucose, mannose, modified glucose. So it's in a certain way, this glucose oligosaccharide is established, and it's first it's linked to a lipid anchor here, dolicol, yes. So, and during the translocation process, once this asparagine comes into the lumen here, it's recognized by the enzymes that transfer this oligosaccharide to the protein, and it's during the translocation process, not on the protein that's already completely synthesized. So it's during this translocation process, the sugar component is added. And this sugar component is very important in the transfer of the proteins during the Golgi and then to the plasma membrane. And another thing is that the sugar component is during this process, it's inside the lumen, inside the Golgi apparatus. Once it will fuse with the plasma membrane, these sugar components will find themselves on the outer side. So these sugar components may be characteristic features of, for example, mammalian cells. They have this cell-cell interaction would be facilitated by these sugar components. Okay, so what you need to know is that it's attached this oligosaccharide during translocation process. Yeah, protection from uncontrolled digest, also some features retention in the ER, but also transfer to specific organelles. So these sugar components experience certain modification during movement through the Golgi apparatus. There might be some sugar components, for example, phosphorylated, removed, added. So this helps also to help for the retention of certain proteins in the Golgi and to locate it at certain places. Okay, so last part, this will be about how vesicles move between ER, Golgi, plasma membrane, lysosomes, and so all this environment. So again, this, I think Dr. Friedenreich mentioned this, from the between endoplasmic reticulum, Golgi, plasma membrane, proteins are moved around in vesicles, so these little balls of lipids. And so there's a movement of vesicles going from the endoplasmic reticulum to the Golgi, through the Golgi to the plasma membrane. And there could also be vesicles moving in the opposite direction. It's called the retrograde transport. So yeah, this is just saying Golgi is a little complex, having like a cis-Golgi network. There would be Golgi would consist of cis medial trans-Golgi would be layers of membranes, yes. And there would be the trans-Golgi network where it's decided whether proteins would go either to the cell surface or they would go to the lysosome, for example, or proteins come via vesicles from outside and fuse with the trans-Golgi network. So it's like a larger, like a Penn station or something, yes. So when we talk about retention of proteins in a specific compartment, it's not more like it just stays there. It may briefly, it may exit the organelle, but it will come back to the original organelle. So it's more a cycling movement going from, say, for example, ER to the cis-Golgi networks and retrograde Golgi to ER, for example. So there are vesicles movement either from one side to the other or in the opposite direction. And this is not random. This is all very controlled because you have endoplasmic reticulum contains certain protein composition. Golgi is the same, certain protein composition. And so this helps to create an identity of these organelles. So one way to, as I said, is to have a signal sequence on the protein. This would be, you want to memorize, or this here, ER retention signal would be KDEL, lysine, asparagine, glutamine, leucine, which is added at the C-terminus of the ER resident protein. So this KDEL sequence is directly at the C-terminus. And so once proteins would be synthesized, imported into the ER, they would leave the endoplasmic reticulum, they would go to the, for example, cis-Golgi network, and some proteins which are secretory proteins, the squares, they would continue their travel outside. But for proteins which are ER resident protein, they would interact with a receptor that will interact with this KDEL or lysine, asparagine, glutamine, leucine sequence at the C-terminus. So here you can see receptors sitting in the cis-Golgi network, looking out for the KDEL at the C-terminus. Once there's an interaction established, it will go back in the retrograde transport, going back to the ER. So this is the way how ER resident proteins are basically maintained in the ER. So it's not like a like it stays just in the ER. It's briefly leaving, and then by signal it's moving back to the ER. So okay. Cycling of vesicles within the Golgi apparatus. Yeah, this is, I mean, is again going either cis-Golgi network, cis-Golgi medial trans-Golgi network, or the other way, retrograde transport here. Going back, this could also be the same mechanism what I just explained to you with the ER resident protein, so similar processes with the Golgi network. There could also be proteins that are damaged, not properly folded, or so they could end up in the lysosome. Lysosome is to degrade proteins in an acidic environment. And so proteins are recognized for this transport to the lysosome. And so this is here with the, so protein, it's in the endoplasmic reticulum, they obtain this sugar chain, oligosaccharide, and this oligosaccharide is specifically modified at the mannose. Sugar is specifically modified by phosphorylation. So there's a phosphate group attached, and that modification is recognized by a receptor in the trans-Golgi network, so this phosphorylated mannose. And so that helps to get this protein to the lysosome. The receptor itself is then detached and is moving back then again to the trans-Golgi network to pick up another proteins with this phosphorylated mannose. So you see here, there is a signal by, in this case, phosphorylation on the sugar component to get proteins in a certain compartment. So besides signal sequence in the amino acid sequence. And so in this case, it's a sugar component. There are, when we go very far, Trans-Golgi network plasma membrane. So we have two processes. One is moving vesicle or material from the Golgi to the plasma membrane, and then it's secreted. So this is like here, Trans-Golgi network, going vesicles, and it's constitutive secretion. So this is just an ongoing process. There's no signal. And in contrast to where components are stored in vesicles, and only in the presence of a certain signal, these vesicles will fuse with the plasma membrane and release the components to the outer side. So this is by, for example, neurotransmitter, neurotransmitter hormones. For example, histamine would be an example. It's stored in vesicles, and only in an allergic reaction you would see release of this histamine in the bloodstream. So, okay. This process is called in overall exocytosis, which means the transport of material from the Trans-Golgi network to the plasma membrane, and then either secretion of proteins to the outer side. The opposite would be endocytosis. This is when material coming from the outer side is brought into the cell. And some well-established example would be import of lipoproteins or for example here, low-density lipoproteins, LDL particles into the cell to help to navigate the cholesterol levels. And so this was also, for example, found by Brown and Goldstein. They got Nobel Prize for this. And so they found there's a receptor that binds the LDL particle. This is internalized by a vesicle. So it will here vesicles formed, and then this vesicle forms with a precursor of the lysosome. And in the lysosome, the vesicle is dissolved, and cholesterol is released, and the receptor for the particle is, before it will fuse with the lysosome, this receptor sorry, is recycled back to the plasma membrane. So you can see there's a cycle of receptor binding to the LDL particle, and then it binding to the LDL particle, coming in, release of the receptor, going back to the plasma membrane. So this would be overall called endocytosis. I just put in this slide here, what I guess many of you have used this mRNA vaccine with the COVID time. So lipid particles, liposomes, packed with the mRNAs would fuse with the plasma membrane. Liposome will be dissolved, mRNA will be released, and then translated, and then portions of the viral proteins will be exposed on the cell surface of immune cells, for example. This is also, so here in the first part would be endocytosis, taking up the liposomes or lipid particles filled with the mRNA. The vesicles itself are not just floating around randomly. This is a highly controlled process. One important feature in this process is that some vesicles are coated with proteins, with specific proteins that help to go vesicles from one compartment to another. And one would be, for example, glycerin-coated vesicles would be one part, one option. So here you can see glycerin-coated. This coat is established during budding off when the vesicle is formed. There would be, I mean, you don't need to memorize this thing. It's just there are different ways of these code. Some are called glycerin or COP1, COP2. So different proteins would be attached to this vesicle surface depending if it's some transport between ER and Golgi, or it's more on the trans-Golgi plasma membrane side. So depending on the location, you would see different proteins attached to the vesicle. So that helps all to go to move the vesicles from a specific compartment to another specific compartment. It's not just randomly floating. And I just put this here at the bottom here because some proteins they call SEC1, 3, whatever, SEC31. So that goes back to budding yeast. And I had this one slide with Randy Shackman, and so they who studied this in very detail using budding yeast to figure out which proteins are needed that the vesicle is going from, for example, ER to the Golgi. If these proteins would be mutated, you would see like accumulation of vesicles in the cell because they would be produced by the endoplasmic reticulum, but they would not fuse with the Golgi, for example. So this is just example how this is studied. Okay. And also this is just to make it clear that the movement of vesicles from one compartment to another compartment depends on specific proteins on the vesicle surface. There would be like a signal protein and a receptor, for example. And don't need to study this here. We say just that you know it's a specific process going from one organelle to another organelle by specific proteins on the vesicle surface. I think we are almost, so this here is just docking or fusing of the vesicle with the target membrane, depends on a interaction of a protein on the vesicle surface and the receptor sitting in the target membrane. So this allows to fuse vesicles specifically with the target membrane. Okay, so this, I mean, this is just like what kind of chapters you could look at. I don't know specifically now, but you can look at any like Albert's, yeah, molecular biology of the cell, any kind of book I recommend. Yeah. And again, this would be example what you will see at the uh, exam. Um, again, 70 questions. So for this part, for example, would be 10 questions. And you will see also, I mean, it's not organized like now mitochondrial secretion, then I don't know what Dr. Freudenreich. So it's all randomly uh, in a random sequence. So it could be from the next lecture and so. Anything on your mind, or it's maybe too much? Yeah, as I told you, study maybe in small portions, like 5 slides, start 1 to 5, then repeat this, go to the next 5. I don't know, maybe anything. Mm. I mean, we can be found on G level, cell biology, if you want. If you have some specific questions, I mean, I cannot explain everything. I'm not just sitting there and waiting for you. But if you have specific questions, you want to stop by for 15 minutes or so, just write me an email and yeah. Okay. Okay. Thank you. Thank you. Thank you.